So now let's discuss the link between ulcerative colitis and cancer. So unfortunately, ulcerative colitis patients have an increased risk for colorectal cancer. It can be up to 2-fold higher. So to remind you all, cancer is a disease where healthy cells transform, start to grow uncontrollably, and spread throughout the body. So on the left here, we see normal tissue, and on the right, we see the cancer cells where there's this uncontrolled growth. And cancer is triggered by genetic changes to the DNA inside the nucleus of your cells. And in ulcerative colitis, there's, as I said before, chronic inflammation, which results in the epithelial cells constantly being destroyed and regenerated. And during these cycles, there's damage to the DNA, and this can trigger the cancer that occurs in the colon. So for patients with ulcerative colitis, it's important that they undergo screenings for cancer. So we know that early detection of cancer leads to better patient outcomes. So here we're looking at statistics from the UK for 3 different types of cancer: lung, breast, and colorectal. And we're seeing that when cancer is caught at the earlier stage, there's much better chance of survival over 5 years, underscoring why we need to really try to capture cancer early. Now, when a pathologist is looking at an ulcerative colitis tissue section that's been stained with H&E, they are looking for signs of dysplasia. And dysplasia is an abnormal growth of cells in your colon that may become cancer. So it's something that needs to be looked at. So on the left here is a colonoscopy, and the gastroenterologist and the pathologist have noted that there's a region here of dysplasia. And this is still pretty hard to see just with a camera. So what happens is they take a biopsy from this section, and a pathologist then stains it with H&E and examines the tissue more closely. And in this darker region of the colon tissue, you can start to see what a pathologist would consider dysplasia. So dysplasia occurs across a continuum in the kind of cancer landscape. So here we have a cartoon of cancer development, where on the left we have normal colon mucosa, and then you start to see that the mucosa starts to change and the structures are really changing, and you go from low-grade dysplasia to high-grade dysplasia, which is more of a risk for cancer, and then finally you have carcinoma, which in this case would be real colorectal cancer. So now let's look at what those tissue histology images would look like from the dysplasia. So on the left, we have an H&E image of low-grade dysplasia, and on the right, an H&E image of high-grade dysplasia. And one thing a pathologist would note for the low-grade dysplasia is you have these hyperchromatic nuclei. So this is a mouthful, but what this means is the nuclei are darker than normal. So remember, the H&E stain stains the nuclei of the cells, this purple-blue, and now these are showing up even darker. And this is often a sign of accumulating DNA in the nucleus, which typically is associated with cancerous cells. In the high-grade dysplasia, the pathologist has noted this loss of nuclear polarity. So when you look at the nuclei, they're kind of scattered a little bit all over the place. So normally the nuclei of the cell is in a specific region of the cell, and during cancer, you start to lose this regulation of where the nucleus is in the cell. So that's the loss of nuclear polarity that a pathologist would note in this tissue section. So what have we learned so far? Well, you've seen a lot of tissue pathology images, and you probably can see that they're incredibly powerful. I mean, you haven't seen enough to probably become a board-certified pathologist, but you hopefully kind of realize that these images are really helpful for diagnosing ulcerative colitis, figuring out what treatment to use, and also detecting cancer early, which is super important. The other thing I want to point out about these tissue pathology images, these H&E images, is that they are very cheap to collect. This is routine care, and millions of these images exist in hospitals. So this is, in a sense, a really amazing data set for machine learning because it's images and you have many, many of them, and often they are annotated. So at the same time, the tissue pathology images don't tell us everything. They don't tell us how ulcerative colitis arises. It just tells us there is ulcerative colitis. So it doesn't tell us anything about the disease mechanism. It doesn't tell us, how a cancer could arise. And, there's a lot of interest in understanding disease mechanism because once we understand how a disease arises, we have pathways that we can start to target with drugs that we could develop. And these are really active areas of clinical research. So if you go to clinicaltrials.gov, you'll see that there's 300 active studies right now for ulcerative colitis and about 2,000 active studies for colorectal cancer. So there's a lot of opportunity in this challenge to work with these tissue pathology images, but ask what more can we learn from them than what a pathologist already learns. And then also, using these images, could we get even better at detecting cancer earlier and impacting patients with ulcerative colitis? So at this point, I've given you an introduction to the biology of autoimmune disease, in particular ulcerative colitis, and we've learned how pathologists can diagnose these diseases, and also how ulcerative colitis increases your risk of cancer and why that means we need better ways to detect cancer earlier.